Good afternoon, everyone. My name is Abbas Roayaei Ardakany, and I'm a postdoctoral researcher at Lahore Institute. Today, I'll be talking about Mustache multi-scale detection of chromatin loops from high-C and micro-C maps using scale-space representation. So, as we know, the chromatin is not folded randomly in the nucleus. The chromatin folding is hierarchical, all the way from the chromosome territory to the nucleosome, and it's associated with transcription. High throughput chromosome conformation capture, HiC, has been the main assay of choice to study the 3D structure of the genome. Significant interactions in HiC contact maps can indicate potential structural and regulatory interactions. For example, you see here multiple domains, some of which are demarcated with convergent CTCF, namely CTCF loops. Loops can be seen as a strong blob-shaped regions in the contact map, and there are methods to find significant interactions in the contact map, which can be classified in two groups. Global enrichment-based methods and local enrichment-based. So the global enrichment-based methods globally fit a statistical model to the contact map data and assign p-values to each individual pixel. So the drawbacks of this group of methods is that they usually don't take into the account the locality information. They assume the interactions are independent of each other. Therefore, they report hundreds of thousands of loops, which is difficult to interpret. So the local enrichment-based methods identify 2D peaks in the contact map, and the drawback with this group is that they usually use fixed size local neighborhood to model the background, which means they might miss loops caused by the proximity of longer or smaller interacting DNA segments. So hiccups in this group has been the main tool to detect chromatin loops. It calls a pixel a loop if it's enriched compared to a set of fixed size local neighborhood. And the other method in this group is SIB, which was recently published, and it's using image processing operations on the contact map, such as Gaussian blurring and morphology to generate the potential loops, which go to a set of post-processing steps and finally gives the final chromatin loops. So we're proposing here Mustache, which falls into this group and tries to address the drawbacks that we mentioned. Mustache is using a theory called scale space from the computer vision to find loops in multi-scales. So as we discussed earlier, loops are reflected as blob-shaped objects with different sizes in the contact map, which correspond to different lengths of the interacting DNA segments and also the strength of the loops. Mustache uses the scale space representation to define the contact map in multiple scales. As the first step, we distance normalize the contact map, followed by a Gaussian blurring, which gives us the first step of the representation. And then we convolve a gradually increasing Gaussian filter with each step and to get the next step. And finally, we get all the Gaussian images. And so then we subtract the adjustment levels to get the difference of Gaussian, which is a good estimate of the second order derivative or Laplacian of the contact map, which we can use to detect the blob-shaped objects in the contact map. So there are some post-processing steps in the Mustache. The most important ones are the non-maximum suppression, in which we find a local maxima of the difference of Gaussian images. So we check each pixel and compare it with the neighborhood at its own level and the level above and below to find a local maxima. And then we have the stability test in which we check each pixel XY to be a local maximum at its own level, at least in 2 consecutive levels, actually. And then we find the connected components for the potential loops that we identified. And then in each connected component, we choose 1 pixel and based on the p-value and call it loop. Mustache finds more loops compared with hiccups and SIB. For example, if we apply Mustache on GM cell line in 5 kb, we are recovering 81% and 73% of the hiccups and SIB loops, while we are generating actually many more loops. Mustache loops are more reproducible. So if we apply Mustache on GM cell line, 10 kb resolution, 2 replicates of the GM, we have a good overlap between the 2 replicates. 74% of one is overlapping 80% of the other, which outperforms the other 2 methods. Then we aggregate the interactions around the detected loops in a 100 kb by 100 kb neighborhood and calculate the APA scores based on the center pixel. As you can see, the loops are enriched, and the APA score of Mustache is lower than the other methods, but due to the multi-scale nature of it. So if we take the 3 by 3 square in the center instead to compute the APA scores, Mustache will get an APA score almost the same as the other methods. Then we take a closer look at the 1 megabase region of GM cell line chromosome 1. At the top, we can see the contact map. And at the bottom, we can see the interactions shown by arcs and tagged by the name of the methods, which can identify that loop. M stands for Mustache, H for hiccups, and S for SIB. So as you can see, Mustache is calling all the loops that the other methods are calling, plus 3 extra, for which we have convergent CDCF on both anchors, which means they are CDCF loops. Then we see if the other data types are supporting the detected loops. So we take CHIAPET, high chip, and promoter capture high C loops as reference loops, and then see how many of them and what percentage of them we are recovering by applying methods on high C data. So the x-axis is the number of loops we are calling in high C data. The y-axis is the percentage of the reference loops we are finding. And the color shows the method. So we did that for 6 different loop sets using these 3 data types. And the blue, which is Mustache, you can see is finding a better, larger percentage of the reference loops, and is outperforming the other 2 methods in all the 6 loop sets, reference sets. And An interesting observation is that Mustache is recovering more high-chip hiccups loops than the hiccups loops in Hi-C itself. In the next step, we check how many and what percentage of the loops are connecting promoters to promoters or promoters to enhancers. And again, you can see the blue Mustache is finding many more loops connecting promoter to enhancer with a better percentage, and that's the same for promoter to promoter. In the next step, we check how robust the detected loops are by evaluating the effect of the sequencing depths. So we subsample the GM cell line data with 3.7 billion intrachromosomal reads in 5 kb resolution to 2 billion, 1 billion, 900 million, and so on. And you can see that Mustache loops are very robust. With only 25% of the original data, we are recovering more than 50% of the loops in the original data. So chromatin folding at the scale of the regulatory elements has been largely unexplored because of the resolution limitation of Hi-C due to the restriction enzyme digestion. So micro-C replaces this restriction enzyme in Hi-C with aminase for digestion, providing the resolution of a single nucleosome to investigate chromatin organization, which enables us to identify functional links between chromatin folding and gene regulation, such as promoter-promoter links, enhancer-promoter links, and repressive patches. So we apply Mustache and Hi-C and micro-C and compare the results. So on human foreskin fibrillous cell line in 5 kb resolution, Mustache calls about 36,000 loops in micro-C and 16,000 in Hi-C. And as you can see, Mustache is calling many more loops in micro-C, which recover almost 90% of the Hi-C loops, which means micro-C not only is finding what Hi-C finds, but many more. Then we compared Hi-C with micro-C with APA plots, and as you can see, micro-C loops have strikingly higher centropixel enrichment in APA plots, which is consistent with previous observation that micro-C enriches the signal to the background ratio. So if we visually compare micro-C with Hi-C, we can see that with the same number of loops called in each data type, we have loops particular to each data type. And for the shared loops shown by a star, you can see the detections are smaller and stronger in micro-C, which is again consistent with what we saw in APA plots. So for the same threshold, Mustache finds many more loops in micro-C, and you can see we are recovering all the loops in Hi-C plus some extra loops. And again, you can see for the shared loops, micro-C loops are smaller and stronger. Then in the next step, we applied Mustache on a high resolution 1 kb on human foreskin fibrillous chromosome 1. So in 5 kb, if we compare Mustache with SIP, we can see Mustache is finding many more loops. And if we apply Mustache on 1 kb, you can see we are finding loops as distant as 1.3 million base pair. And at the same time, if we zoom in on a small region of this, we can see Mustache is finding loops as short as actually smaller than 20K base pairs. which has been very challenging due to the limitation of high-C to date. So with that, I would like to conclude my presentation. We presented Mustache, a new method for finding chromatin loops in multiple scales. Mustache finds more reproducible loops than other methods. The detected loops are supported by other data types, such as ChIP, high-chip, promoter capture, high-C. Mustache results are highly consistent between micro-C and high-C data of the same sample. Mustache takes less than 2 hours to run on human data genome-wide. Mustache is scalable and efficiently runs on high resolutions such as 1 KB, 500 base pair, and so on. And Mustache is available on GitHub. Please get it to try. Thank you all for listening. I'll take questions.